In the ongoing lecture series on high throughput screening and its role in natural products, we have already discussed that what is HTS or high throughput screening. We also discuss about the brief historical development of this important technique that is being used for drug discovery process. We also discuss about different types of HTS in terms of in silico, in vitro and in vivo methods. We discuss few of the in vitro assay techniques such as fluorescence resonance energy transfer, that is FRET, or time resolved fluorescence resonance energy transfer, that is TR-FRET, then alpha screen assay, alpha LISA assay, scintillation proximity assays, SPAs which we call them. Then we also discuss about the reporter gene assay, what are their applications, what are their limitations, and in the end we discuss high content imaging technique that is now being used for high throughput screening. So friends, in continuation with the ongoing series, I will discuss today another important technique of HTS that is three-dimensional cell culture. Now in the recent past, or I'll say up to recent past, two-dimensional cell cultures were widely used by pharmaceutical industries or research organizations in the drug discovery process. Now if we talk about two-dimensional cell culture studies, it's nothing but a monolayer of cells that is being grown under in vitro conditions, and various drugs are tested on those cells. But the major limitation of two-dimensional cell culture was that because in a normal living system, cells are surrounded by the cellular matrix, they also interact with the neighboring cells, and such kind of interaction, such kind of effect of the matrix on the cellular physiology was not being taken account in 2D cell cultures. And many a times it has been seen that certain drugs which were found very effective in two-dimensional cell culture studies were later on found to be inactive, and vice versa also. There was a study carried out wherein an extract of a plant was evaluated for its anti-cancer potential by using two-dimensional cell culture studies, and it was found that the compounds which were present in the extract were inactive. But when that extract was then evaluated using a three-dimensional cell culture, then the efficacy of a compound was found, and from there it was assessed that if we would have been using a two-dimensional cell culture, that compound would have been missed. And therefore, three-dimensional cell cultures are now being used for high throughput screening in drug discovery process. So we can define a 3D cell culture as a culture environment that allows cell to grow and interact with the surrounding extracellular framework in 3 dimensions. Now when we talk about extracellular framework, it means the cellular matrix which is in which the cells are present, vis-a-vis also the neighboring cells and how the cells are interacting with those cells, what kind of effect is matrix is having on to the cells. Now if we have a brief comparison of 3-dimensional versus 2-dimensional cell cultures, we can compare them in terms of different parameters. First one is the morphology. As I told you that in 2-dimensional cells, they were largely present in a monolayer, and therefore they have a flat sheet like appearance, whereas in 3-dimensional cell cultures they have a natural shape in the form of spheroids or aggregates. And if we talk about proliferation of the cells in 2D and 3D cultures, then in 2D the cells normally proliferate at a faster rate than under in vivo conditions, whereas in case of 3-dimensional the rate of proliferation is a bit slower, or it can be faster depending upon the type of study you are performing. So it varies depending upon the type of culture in comparison to the 2-dimensional cell cultures. Now if we talk about the exposure to the drugs what is the difference between the intensity of the exposure to the drugs in case of 2D and 3D cultures, then as we discussed that in 2D cultures, cells are normally present in a flat sheet like arrangement forming a monolayer. Therefore, it is believed that all the cells are equally exposed to whether they are drugs, whether they are nutrients or any kind of growth factor that has been present in the media. Whereas in case of 3-dimensional cell cultures, they may not be able to fully penetrate the spheroid. It may happen that those nutrients or drug may have an impact on the outer core of the cells or outer region cells, but they are not able to penetrate into the central core of that spheroid. So, we can say that the cells are not equally exposed to the drugs or to the nutrient media or to the growth factors, unlike 2-dimensional cell cultures. Coming on to the difference on the basis of stage of cell cycle, in 2D cultures, most of the cells are likely to be in the same stage of cell cycle, whereas in case of 3-dimensional cell cultures, since there is a different rate of proliferation, there is a different ability of the drug penetration to the cells. Some cells would have major exposure to the drugs, whereas some cells will not be able to have any contact with the drug, and therefore such kind of arrangements in 3-dimensional cultures, they have cells in different cell cycle stages. There may be cells which are proliferating, there may be cells which are under dormant conditions, there may be cells which are hypoxic, or there may be necrotic cells. So, that is large variation in case of 3-dimensional cell cultures, whereas in 2D cells are present in almost the same stage of the cell cycle. Now, if we talk about the difference in terms of gene expression or protein expression, then it has been observed that 2-dimensional cells under 2-dimensional cultures, they often display a differential gene and protein expression levels in comparison to the in vivo models. On the contrary, the cells that are present in the 3-dimensional cultures, they often exhibit a similar kind of gene and protein expression profiles in comparison to those of the in vivo tissue organs, because these cells in 3-dimensional are present in almost the similar kind of natural environment in which the cells are present in the living system, and therefore their gene or protein expression profiles are almost same what we can observe in the in vivo tissues or the tissue organs. In terms of drug sensitivity, then cells, they are more vulnerable towards the treatment, and drugs, they basically appear to be very effective in case of the 2-dimensional cell cultures, whereas in case of 3-dimensional cell cultures, cells are more often resistant to the treatment in comparison to those of 2-dimensional systems, because again we have different types of cells present. We have a spheroid arrangement of the cells, so the inner core cells may not be are not able to get exposed to the drug. Therefore, 3-dimensional cells, they present a more appropriate picture in terms of the effect of the drug, and that picture is more relevant or is more comparable to the effect that are observed when those drugs are tested under in vivo conditions. Now, after discussing the comparison between these two types of cell cultures, I will just highlight some of the advantages of 3D cell models or 3D cell cultures in case of high throughput screening. So, coming on to the first major advantages, since these cells are present or in an environment which mimics the environment of a natural natural conditions of the cell present under in vivo conditions, and therefore 3D cell cultures offer us enhanced productivity. And when we say about productivity, it means we can very well predict the behavior of the drug in terms of its efficacy, in terms of its toxicity, and such kind of enhance predictivity in terms of the effect of drugs. It also helps in reducing the overall cost of the process, because many times it has been seen that once the drug has been found effective, but later on when that drug is being tested under in vivo condition, that drug molecule was found to be toxic. So, whatever investment was done, whatever labor that has been put in while evaluating that drug molecule under in vitro or two-dimensional cell culture studies, that all goes waste. Second major advantage is, since these cells that which are present, as we discussed, that these cells are normally present in an environment which mimics the exact conditions that are present under the in vivo conditions, and therefore the results of the 3D cell cultures are more realistic, and they very well give a very good idea about the penetration abilities of the drug, or in simple terms, if I say drug absorption characters can be easily studied by using 3D cultures, vis-a-vis also the distribution pattern of the drug. Then the third advantage is the 3D cell cultures. They basically help in understanding the complex processes that are going on inside a biological system. Since again the same reason, these cells are present in a environment which is almost same as that of the in vivo conditions, or the cells which are present under in vivo conditions, and therefore such kind of a system also helps in understanding certain complex processes, physiological processes, biological processes that are going inside the cell. These cultures also help in studying long term effects of the drug, and for such kind of studies normally longer culture durations are being used, which also helps in understanding, or which also helps in evaluating the effect of drug under chronic toxic or chronic toxicity conditions, or the long term toxicity effects can be easily assessed by using 3D cultures. Then it also helps in reducing the usage of animals. Since we are providing the environment to the cells same as that that is available under in vivo conditions, it also helps in reducing the usage of animal, and thereby reducing overall the animal testing process. Now, I will just briefly give you an idea about the protocol that has to be followed while using high throughput screening. So, now till now we have discussed about HTS, its classification, different assays, whether they are in vitro or in vivo, that are being used to evaluate the potential drug molecules or the lead molecules. Now, how to follow that process, what protocol we should follow. So, the first and the foremost step is the selection of the screening method, whether you are going for a target screening or you are going for a phenotypic screening HTS method. So, depending upon the requirement, what kind of results you are expecting from your study, first and foremost step is you have to first select whether you are going for a target oriented HTS screening or phenotypic oriented HTS screening, and both these types of screening methods we have already discussed in my previous lecture. Next step is the development of the assay. So, what kind of activity you are going to evaluate, for that you have to develop an assay. Not only you have to develop the assay, but also you have to optimize that assay in terms of the usage of the reagents, in terms of the usage of the cell density, in terms of usage of the incubation period, pH of the nutrient media, and so on and so forth. There are so many parameters which you have to optimize with respect to your assay. And then once you have optimized, then you also have to validate whether the assay which you have developed is specific to that particular disorder which you are evaluating or not, because if it is not specific, then the whole process goes in vain and you are not going to get the exact results which you are looking for. Once the assay method has been developed, optimized and validate, then the next step is you implement then that assay in terms of evaluating the test drug molecules which you want to evaluate. Once the HTS has been implemented, then you get tons and tons of data. That data is acquired by using suitable detectors, suitable equipments, then data is then further analyzed and interpreted, what kind of final results you have obtained. And last is, once that data is analyzed, then you are left with either hits or the lead molecules, and you can process this hit or lead molecules for the further drug development. Now we move on to the role of high throughput screening in natural products. Now we know that HTS is a technique that was being largely used for synthetic molecules, but in the recent past we have seen that HTS is now gaining interest in terms of the natural products as well. So I have just displayed a graph wherein the data of last 25 years is being presented. When I search the PubMed using a keyword high throughput screening, there were around 53,000 hits in terms of the articles, review articles, book chapters, and so on and so forth. Now, when I use a keyword that high throughput screening along with natural product, the number drastically decreases to 2,718, approximately 3,000. So we can say that there was approximately 0.5% of the publications that were mentioning the usage of high throughput screening for natural product. Though there is an increase in the usage of HTS for natural products as well, but still the figures they show a very meager picture of approximately 0.5%. What are the various reasons why HTS is now not being so widely used for natural products? We are going to discuss ahead. Now, if we look into the libraries that are being used in HTS for natural products, as we discussed that largely this technique is used for synthetic molecules, and various libraries of synthetic molecules have been developed which consist of hundreds and thousands of compounds which are evaluated for a specific disease or specific disorder using HTS. So, in case of natural products, there are different types of libraries that are being used. First one is crude extract library. As the name indicate, these libraries are nothing but these are the number of extracts that are being prepared by using different plants. And we know that since these extraction processes are simple, you can simply extract a drug with a suitable solvent and you get the extract. So, these techniques or the methods which are used to prepare these libraries are simple and they have a lower initial cost. Second is the pre-fractioned libraries. In such kind of libraries, the extracts which we have prepared, we fraction those extract in different fractions, whether in terms of usage of solvents in increasing order of polarity, so that we can differentiate the extract into fractions of different solvents in increasing order of their polarity. Or we further purify the extract by using appropriate method and we get a suitable fraction that has a highest concentration of the required constituents in it. So, we basically by purification we are removing the interfering compounds. So, such kind of libraries, since an additional step of purification or fractionation is added, then these are a bit of higher cost in comparison to crude extract libraries, but nowadays major focus is on pre-fractioned libraries, and as we will discuss ahead, lot of databases or the research groups or research institutes, they are mainly focusing on the pre-fractioned libraries. And third one is the pure compound libraries, wherein we isolate the compounds from extracts or fractions, we characterize those compounds and then those compounds form a part of the library. So, we isolate and characterize so many compounds from different extracts of different plants and then we prepare a library. Since it involves the usage of various chromatographic techniques in after extraction and fractionation, so the cost of developing such kind of libraries is huge in comparison to extract or pre-fraction libraries. One. Second, development of these libraries take hell lot of time because the chromatographic separation and characterization itself is a monumental task in terms of identifying the compounds or isolating pure compounds, and therefore majority of the research institutes or research organization, they are having very small number of pure compound libraries. So if we look into the flow chart of the usage of HTS in for natural products, it has around 6 steps, major steps I will say. Then first one is the selection of the plant material. Second is you extract that plant material by using suitable extraction technique, whether they can be modern techniques by and large which are used, supercritical fluid extraction, ultrasound assisted extraction, microwave assisted extraction, and so on and so forth. Third step is automated fractionation of those extract. So once we are dividing those extracts into different fractions, then fourth step is the concentration of those fractions. These fractions are automatically evaporated. The dry powder that is obtained is then again weighed. It is again weighed automatically because HTS is a fully automated process. So all the fractions that are obtained after evaporating the solvent, they are dried, they are weighed, and then they are labeled by a proper coding technique by which we can scan the code and we get all the information regarding that fraction. Now this is the natural product library sources which I was talking about. There are few institutes, and you will see that majority of the institutes, they have higher number of pre-fractioned libraries in comparison to extract, barring one or two institute. And you will see that majority of the institute, they have a very less number of pure compound libraries. As in this table we can see analytical quant discovery in Germany and US, they are having around more than 25,000 semi-synthetic compound library, and if we talk about the pure compounds obtained from natural products, it's merely more than 5,000 compound library. Then here also we can see that all the institutes, they have higher number of fraction library. Griffith Institute is having 1,12,000 fraction library. Phytopharmacon in USA, they are having around 25,000 fraction library. Then Development Therapeutics Program, that is the National Cancer Institute USA, they are having 3,26,000 fraction library. And if we compare to the number of the pure compound libraries, for Phytopharmacon it's only 500, then 69,000 for InterBioscreen because they are not dealing with extraction fraction, and merely 419 pure compounds in terms of the National Cancer Institute USA. So that's why more and more focus is now on developing the pre-fraction or the fractioned library, that fractions are obtained from nowhere from but crude extracts. Now despite there, as we discussed, there is an increasing interest of usage of HTS in high throughput, increased usage of HTS for natural products. But there are few challenges why we cannot or why the usage of HTS is not to a full scale for natural products. And what are those challenges we are going to discuss in the next part of the lecture. Thank you.